Okay. Hi, everyone. Welcome to the R for Bioinfo session. So today we have the great pleasure to have with us Xueyi Dong. Today's session will be about RNA-seq, and we will show you how RNA-seq is as easy as 1, 2, 3 with limma, Glimma, and edgeR. So I'm Hedia Tani. I'm postdoc at Pasteur Institute of Tunis at the Bioinformatics Lab. I'm also co-founder of rstats Tunis, and I will be the host of this session. So I will present Xueyi Dong. Thank you, Xueyi Dong, for being with us today. Xueyi Dong is a PhD candidate at Walter and Eliza Hall Institute of Medical Research, Melbourne, Australia. She loves to share her experience with RNA-seq data analysis. Xueyi Dong has translated the analysis workflow package. So there is a package called RNA-seq 1, 2, 3 in the bioconductor into many languages and as Mandarin. And also she taught this, this kind of workshop on different events such as BIOC Asia. Actually, I assisted to one of these events and to the BIOC Asia 2020 event where Xueyi Dong presented a workshop about RNA-seq 1, 2, 3 in Mandarin. Thank you, Xueyi Dong. So before letting letting Xueyi Dong introduce you to the RNA-seq 1, 2, 3, let us present you and introduce you to the Our Ladies and Our Ladies Tunis initiative. Mona, please.
Okay, thank you, Hedia.
Before starting the workshop, we'll introduce you to the Our Ladies Global and Our Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of Our Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Caros founded Our Ladies on October 1, 2012. Our Ladies Global was born and the grant was awarded in September 2016. Since then Our Ladies has grown to 170 chapters in 44 countries and 39,000 members. Our Ladies Tunis is part of Our Ladies Initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of Our Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop.
Thank you, Monet. So thank you, Xueyi Dong, for being with us today. I attended, as I said, the BIOC Asia 2020, and the workshop was actually in Mandarin. So let's have the joy and the pleasure today to have this RNA-seq is easy as 1, 2, 3 with limma, Glimma, and edgeR in English. So Xueyi Dong, the screen is yours.
Cool. Hi everyone, my name is Xueyi, and although I'm from China, but now I'm in Melbourne, Australia, and yeah, and I'm a PhD student at Xueyi in Melbourne, Australia, and I will teach about a workflow that can make your RNA-seq analysis easy as 1, 2, 3 with our packages. And first of all, I will suggest the participants to install the package called RNA-seq 1, 2, 3 it's a bioconductor package, so you can. I will give you a few seconds to install to input the code and to install the package. Or maybe I can copy the code into
we'll
the.
put this in the chat. So we.
I can also put it in the chat.
Okay.
See how can I do this? Okay, it's not easy for me to open the chat. So let me see. Yes. Okay. So you can copy the code in your R or RStudio and to install the package. And while you are doing the installation, I will continue to my introduction to RNA-seq. So first of all, we will start from some basic knowledge about. Let me see if it's. Some basic introduction about what is RNA-seq. So okay, is this the right screen that's showing the slide?
We have introduction to RNA transcriptome
cool.
and RNA-seq.
I'll go on. So in our body, there are a lot of kinds of different cells, and different cells are looking quite different, like the nerve cell in our nerve system or the muscle cell. Well, however, all the DNA sequences in all these kind of different cells in one human, or we say in one other organisms like in mouse, all of the cells are having exactly the same DNA sequence. But why do the cells look quite different? This is because different genes are turned on or turned off in different cells. Like in nerve cells, some genes are specifically turned on, while they are in muscle cells, they're turned off. And the same thing also happens reversely. And once the DNA is turned on, what will happen is that these genes are the protein are the thing that actually usually actually controlling our body, but the DNA the genes are the periods of DNA that have some biological meaning that and the genes actually coding the protein. And from DNA to the produced protein, firstly the gene will be transcribed to mRNA to RNA. That we use the gene as the template, and the mRNA we can consider as copied from one chain of the DNA molecule, and then the RNA will be translated into protein. And other than mRNA, there will be also some other kinds of RNA, like non-coding RNAs or tRNAs or something that they didn't produce protein, but they also have some special meaning to our life. And to be more specific, the gene is not only it's not only having the coding region of the that we may they may produce a mRNA and it can be translated protein. Actually, there are some region in genes that we call intron, and after the transcription, we are only copying the whole gene to some form of mRNA, but it's not mature. Then when we have this, there will be some splicing of the RNA. So the intron will be cut out from the RNA. We can consider like the RNA are cut into different pieces, and some of the useful pieces will be linked together again to form the mature mRNA. And after it gets mature, nature, there will also be a poly A tail to be connected to the end of the gene. The end of the mRNA. And after that, the mature mRNA can get translated, and there will be some mechanism that helps it to produce the protein from the mature mRNA. So RNA is very helpful. To study RNA is very useful in our biology studies, and currently there is a subject called transcriptomics. That's the study to study the transcriptome. Well, the transcriptome is defined as the collection of all of the transcript readouts present in a cell, or we can say that it's all of the mRNAs in a cell. And transcriptomics is to study them in a whole. So it's like study all of the mRNAs present in a cell as a whole. And there's a slide about the brief history of gene expression technology that, and from the very beginning, if we want to study transcriptome, people use something called microarray. That it is a small chip, and on the chip there are a lot of different places that would have some specified adapter design that they can recognize different RNAs, but however the technology is limited because the places on the chip that you can identify RNA is very limited. And if you want to study a human sample, you have to have about more than 10 chips for each sample. And for doing that, you will need quite a lot of the RNAs, the RNA materials to do the experiment. And sometimes the material is not enough and it can cause trouble to the study. Well, currently, most of us are using RNA sequencing to do the study. It's some more advanced way to do the gene expression analysis, that we sequence the RNA in the cells. And what can we do with RNA-seq? Well, it can actually be used to explore or quantify the transcriptome of an organism, which can be utilized for the following types of experiments. First of all, is differential gene expression, that we evaluate what gene is changing its expression between different groups of cells. Like if we have normal cell and if we have some normal samples and we have some samples like having cancer, and so we can compare between the normal and the cancer cells to see what gene is turned on or specifically turned off in the cancer cell when we compare to the normal cells. And another thing we can do is to do a transcriptome assembly. That is, we can build a profile transcript region of the genome. And also we can use RNA-seq data to help build a better gene model. That's sometimes we know there is a gene on the genome, but we don't know how does it look like which part is an exon. That's the part that is kept in the mature RNA, and where is the intron? That's the part it was spliced out when the RNA is going through the splicing. And also there are some other things we can do, like a meta-transcriptomics or some other analysis. And today we will focus on the differential gene expression analysis. Maybe this is the most useful, not the most useful, but the most commonly used thing with RNA-seq. And let's go through the mechanism of RNA-seq. That for the library preparation, firstly, is the first thing we want to do is to extract the RNA from our samples. Sometimes we just extract the mRNA, but sometimes it's the total RNA, all of the RNAs in the samples, and like it can contain some non-coding RNA or like tRNA. That's the thing that helps the translation. And after that, we can use some kinds of enzyme to remove the contaminant DNA. But sometimes if we want to extract the RNA, some DNA can also be extracted falsely. So after removing the DNA, we will select the mRNA, because in human there are only 2% of the RNA are actually the mRNA, and I believe most people are mostly interested in studying the mRNA, but not the other things. So we can either remove rRNA or select mRNA. Well, actually, the most times what we do is to select the mRNA, because I said that the mature mRNA have a poly A tail at the end, and we can use the poly T, which can be matched to the poly A, can be attached to it, so we can select them. And during this step, usually the RNA will be fragmented. Like if we use some physical way, or sometimes using some non-specific enzyme to digest the RNA. Because the machine, the sequencing machine we usually use for next generation sequencing, it's the most useful, most widely used sequencing technology at these days. Yep. Because it has a limitation that it cannot, the length of the mRNA fragment it can detect is very limited. So usually we will do the fragmentation of the RNA. And because RNA is usually very fragile, that it can be if the condition is not very good, it may break. So usually we do the reverse transcribe. So we have the cDNA, and usually we'll also have the to make it into a double-stranded cDNA. So it can be more, it will not be that fragile, and it can survive in different conditions. And after that, we will have the sequence adapters to ligate at both of the end of the cDNA. And once we have the adapters, and sometimes we will also do some PCR, is to amplify the signal so that we can have more materials to detect. And after that, we will select the size of the fragments to make sure it can be sequenced. And so that's the library preparation. And after getting the library, we can have the site-selected cDNA with sequencing adapters like this. And there are two types of technology. Like single end is to only have one read from one direction. While if we do the paired end sequencing, we other than the read one, we also have a read two that is on the reverse strand and on the reversed, and it's at the other end of the pair. So like sometimes if the read can be mapped to a unique place on the genome, then it's fine to have the single end. While sometimes there are a lot of regions on the genome that looks very similar, and it's not easy to just mapping one little piece of the sequence. So maybe we can. So if we have paired end, we will have two different pieces. Then we can map them. And if they can be mapped to the same place, that we can know it's the right place. So it can help the mapping. And for some of the analysis, specific analysis, like if we want to study the splicing events, that we want to know some information about the junction between splicing junction, which means the border between one exon and another one exon, then it will also be useful to use the paired end. Yeah, and of course, paired end will cost more money to do the sequencing. So I think single end is already enough for a lot of situations. And there is a video to show you how does it do for the how can we sequence them.
Good.
Video.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. For reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are chromally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the read 2 sequencing primer. As with read 1, the sequencing steps are repeated until the desired read length is achieved. The read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base pairs are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely tracked.
Okay, so that's enough to introduce the mechanisms of Illumina sequencing. And let's have a short break, and maybe we can be back in 5 minutes. And if you want to view some of the materials about the introduction, actually I use some online resource from Harvard, and you can go to this website. And if you wanna have a look through the workflow I'm going to introduce, you can go to this. Or if you want to learn some math related to the analysis, which is basically about the introduction to the some linear models and different designs of the how to treat the different designs of the study, how do we how to build a design matrix, you can have a look at this. And if you have any question, you can type into the chat. And
Okay, so let's have 5 minutes of break and we'll put all these links in the chat.
I'll be back soon. Okay, so it's time for us to start our data analysis workflow. And there's some basic introduction about RNA-seq that I just talked about. But to emphasize it, I will repeat it again that RNA-seq is a method that we can quantify the amount of RNA in a sample. And actually, we can also do some other things. But today we are gonna use RNA-seq to measure the gene expression level, so we can know which genes are active and how much they are transcribed. And the differential expression analysis is the analysis that we compare the gene expression between different groups. How to say we want to find out which gene are changing their expression between different groups. For example, if maybe we want to compare normal cells to the mutated cells, or we want to compare the treated cells to the control cells. And after getting the raw information from the RNA sequencing, the data looks like quite a lot of short pieces of the RNA sequence. And yeah, actually, since the RNAs are all fragmented and they are just raw sequences containing A, C, T, G or something, that if we just directly look into the sequences, it's meaningless because we don't know, we don't understand what's this and where does it come from. So usually after some very basic quality control of the sequences, what we do at the first is to map the reads back to some to the genome that we know. Usually we call them the reference genome. We can download them on some datasets, some databases online, and it's usually easy to find, for example, especially for human or mouse. And for some of the other organisms, we can also try to find the reference genome online. So after aligning the reads to the reference genome, then we will know which gene, or to say which region of the genome does the gene come from. And then we will also need some kind of file called annotation file that containing the information of where does each gene located in the genome. So when we're comparing them to our aligned results, we can know which gene does each read come from. And then we can do the quantification of reads, that we can count the number of the reads. We can count the number of reads that are mapped to each gene. And after that, we can get a table. We call it a count matrix, or so in the count matrix, it's a table that each row, each column is a sample in our data set, and in each row, it's a gene, a specific gene, and the number means the number of reads that is mapped to this gene. And for an overview of the RNA-seq, our data analysis, firstly, after the sequencing, we can get the sequenced reads, which is in the FASTQ file formats. And after getting it, we will do the align them to the reference genome. And I would suggest you a package called rsubread, because for a lot of aligners, we will need to use them in the Linux, to use the command lines to run the software, while rsubread is built in R, and it's actually a wrap of a package called subread. So you can run all of your data analysis workflow in R easily. But I don't suggest you to run it on your own computer, because for mapping, it's very a hard task. It will use a lot of the computational resources. So it requires, like, your computer must have a lot of memory and a lot of CPUs. So usually we will do this step on the server, and I will not show you how to run this today, but you can find it in the user menu. It's not very hard. And after the alignment, we will count the number of reads mapped to each gene. And after that, we will get the count matrix. And for doing this step, I will suggest you to use a function called feature counts. It's also in the package rsubread. And of course, for the alignment or count, you can use some other tools. There are also some other very good tools you can use, but I will not introduce them today. And the workflow we are introduced today is starting from the count matrix. After that, and after getting it, we will do some data preprocessing to make the data available to use in the downstream analysis. And we will do the actual differential expression analysis using some linear models. And and the tools we're using.
I have a question.
Yep.
So Abir is saying that he has question about design of the study. So he says, if we are comparing wild type and knockout cells in vivo, so I have a question about design of the study. If we are comparing wild type and knockout cell in vivo, what are we expecting from the results? Just confused, because already we have the gene that we assume has effect knocked out.
Mm-hmm. Yeah, sometimes if you mean you wanna compare the samples with a gene knocked out and the other samples, right? Yeah. Usually, if you knock out a gene, and there will be some because the genes are usually in some kind of pathways. So if one gene is out, it will also affect quite a lot of other genes. So you may see some other genes are also down or up because of the change of one gene, right? Yeah, and because sometimes before the experiment, we don't know actually what will happen, and the data will tell us. But usually, if the data quality is good enough, we can use the analysis to see what we are expecting, is our hypothesis right or not.
Thank you.
Okay. And for the differential expression analysis, the tools we are using are edgeR and limma. And actually, today we are using edgeR to do some data preprocessing. And for the linear model part, we will mainly using limma. And also, if you just using edgeR, it can also do the differential expression analysis on its own, and it's also a very good tool. And also some other tools like DSeq tool are also very good. If you are in favor of the other tools, you can also have a try. But today we're just talking about using edgeR and limma. And after talking about the overview, I will give you some instruction of experimental design that, for example, if you wanna do some RNA-seq experiment and you are designing it, it's very important to know some points. And the first point is that you must have biological replicates. Like if, for example, if you want to compare normal cell to the treated cell, and then so for both normal and treated samples, you will need to have at least 2 samples in each group. And it's, of course, more sample is better, because if we don't have any replicates, it's hard for our model to know whether the difference is from the random thing like the some random error, or it's the real biological difference. So you need to have replicates. And also, if you wanna sequence your samples from one, like if you have a very big experiment and you wanna sequence them in different sequencing runs, then you need to have some reference samples between different sequencing runs. Yeah, like if you're having a lot of samples and a lot of sequencing runs to be analyzed together, you will have you need to have like for each different tool, like the run one, between run one and run two, you will need to have at least one sample sequenced twice in the different sequencing runs. Because when we're doing biological experiments, sometimes some little change of the condition of the experiment or the environment, some very small effects can affect quite a lot for the results. And the difference between different runs, because of the different condition or something we don't know between the different runs, it's called batch effect. And it is actually a very important thing that if there is batch effect, usually in the analysis we'll try to remove it, because it's affecting the, like, for RNA-seq, it's affecting the gene expression, but we don't want to calculate. We don't want to like treat the batch effect as the real biological difference, right? So it's very important to have some reference samples between different sequence runs. And also, like, if you don't sequence all of one group on one day and all of other group on another day, which is similar to what I just talked about, because this will cause some batch effect. And if we don't have any reference sample across different batches, we cannot remove it in the linear model. So it's better if we have two groups and want to sequence them on 2 days. So we can sequence half of the group 1, half of the group 2 on day 1, and on day 2 we can sequence half of the data. And for other potential batch effects like sampling date and sequencing date or technician, who is doing the collecting, who is preparing the sample or something, you need to consider carefully about those things that may cause batch effects. And if you think there is something may cause batch effects, you need to have some reference samples. It's very important because I had some experience with some datasets which are not good, which are very badly designed, and I cannot get rid of the batch effect, and it's a very bad thing. Okay, and we were going to talk about the data we are analyzing today. It's from Sheridan et al. From a paper in 2015, and in our data set, all of the samples are from mouse memory glands, and we have 3 different cell populations. One is called basal, and one is called LP, and one is called ML. We don't need to know much about what exactly are these samples, but you need to remember that we have 3 different cell populations, and in each population we have 3 replicates today. And these samples are sequenced in 3 different sequencing lanes. We can see they're sequenced in 3 different batches. Well, it's not one sample alone, one group alone. It's actually mixed. So we can get rid of the batch effect, and you will see later on. And what we want to know is that we want to compare the gene expression level between cell populations. So that's the data we're gonna analyze today. Okay, and let's get started. So we will switch the screen to show you my RStudio. Studio. Okay, so I'll create. And you can also open your RStudio so you can do it while follow me while you are listening. And actually the workflow we're gonna talk about is also available online, so you can see it in the package RNA-seq 3. Okay, what we will do now, I will suggest to you to in your RStudio choose file and a new project. Click this. And this will create a new R project, which will contain the code we're using and the data. And if
Xueyi, sorry, can you make your screen bigger?
Is it very small now? Yeah, I can try because my monitor is a 27 inches wide, and maybe everything will show smaller on your screen. So let me switch it. Studio. Is it better now?
Yeah, quite better.
Okay, so choose file and new project. Ignore this, and you can either choose the existing directory or create a new one. I would rather choose new one and click this new project. And we will need to have a directory name, which can be RNA-seq 1, 2, 3, or anything you wish it to be called. And you can choose somewhere that you want to put folder in. And let's click the Create Project. Okay, now you will see you have created an R project. So if you wanna switch between different R projects, it's very easy. You can click here on top right, and you can like these are different projects I'm having. So you can click one, and you will all of the files you were opening will be shown here, and the environments will also be there, and so it will be very easy. And at the start, I will suggest you to see this. There is a function called browse vignettes, and you will type our package if you have downloaded RNA-seq 123, and let's run it. And you will see, you will open. There will be something open on your internet browser, like is it showing? And so you will see this. This is a website that have some links to our vignettes. So if you are interested, you can click the HTML of the English version of the EZS 1231. So this is our full workflow. Yeah, you can see it like this. So there are the code and also the results of running the code here also. Also some introduction which are written. So you, after that, you can also read through the article in this way and go back. And if you want to see the source of our code, you can click this and it will be downloaded. So you don't need to type the code I will be showing you today. And also you can click the source. It will be our markdown file that I believe you are also all familiar with, so I don't need to introduce. And okay, let's go back to the RStudio. So first of all, okay, go to, and then we will create a new R script, and we can type our code here. And first of all, that we will need to load the packages we will be using. So using library limma, and you can, if you're using Mac, you can click Command and Enter to run the code. If you're using Windows, you can use Control and Enter to run the code that your mouse is on. And then library Glimma and library edgeR. Some up some of the it's helping. And also another package library. Musculus, it's mouse, it's the annotation of the mouse, which you will use later to annotate the dataset. Okay, done.
Xueyi, Miriam is asking if when we load the library RNA-seq 123, we don't have all these loaded. So do we have all, it's like these libraries loaded when we load library RNA-seq 123?
Good point. I haven't tested, but I believe yes. If it's not, you may need to load them one by one. But usually, if the maybe I can try it and see. If I try library, yeah, because it's a workflow package. It's not like the usual package in R. Oh, cool. Yeah. Yeah, I believe it is actually, yeah, it's loading all of the packages. So maybe we can do this instead of loading one by one. But sometimes, like if you don't wanna load this, then we can load one by one. Otherwise, you can just load the library on sequence 3. It's easier. And then we will, I can copy the code because it's hard to type here. And then we will download the data. And we will copy and paste the code in our chat. And then we will download the file the data we're using. And don't be worried because it's just a small file. So we are downloading it from Geo database, and it's a compressed file having a lot of different files, and each txt file is it's a count matrix from one sample. And now we have the files so that because in this dataset it's having quite a lot of sample, but we are not using everyone. So we are choosing the 9 samples ourselves to be analyzed today. And then use this, we will uncompress the files. Okay. After uncompressing them, and then we will use the files so we'll know which files we are using today. So we are giving the files the different file names we are using today. Okay, and let's have a look. One. That's to show the first 5 rows from the first file. And now you can see here, that's the result. In the first file, the structure of the file looks like this. The first column is the entrance ID of each gene. That's some of kind of form to code the gene. And the second column is gene length. And the third column is count. The gene level count is the number of reads mapped to each gene. And what's mostly used for is the entrance ID, that we can know what gene it is and count. And then we will read the file formally into our studio, read in the data. And we will use this command called read DGE, and have files, columns, to 1 and 3. So that's it. We will call the function. We will call the function, and it will read in our files. What files we're reading is here. These files in the files, and the columns representing the name of the gene and the count is column 1 and column 3. So it looks like this, and we run it. And and our data will be saved in an object called X. And let's see it. the class X to see what kind of what is the structure of X now. So you can see it's something called DGE list. So that's the kind of object we're using in edgeR and limma to store the and it's in edgeR to store the differential expression the RNA-seq data. And let's see what's in R. We can type R. Type X and enter and here, and you will see it's an object of class DGList. And the first data frame inside is called samples. And currently the data frame sample is only having file names, and its group is all 1 because we didn't assign a value to it. And the library side, it's this sum of the total read counts from one sample. And normalization factor are all 1 because we are not calculating. So, and after that we'll have a data frame called counts, the gene level counts. And now we are reading all of the files into our R. And now you can see it's one data frame that each column is a sample, like this sample, this sample, this sample, and each row is it's called text, but actually you can see it's genes. The name text comes from the microarray because Glimma was designed for microarray at first. And in microarray of the position where the gene can be detected are called tags. Actually, now we will know that they are actually genes and not tags. But it's not very different. So let's continue. We will have a look of the dimension of X. So here we have 9 samples, and in each sample we have 27,179 genes in each sample. That's the total number of genes in the dataset. Okay, so our raw data are already read into R. And the next thing we will do is to organize sample information. That's because currently we can go back to here the outputs from X. The samples data frame only containing the file name and library size, but we still don't know which samples come from which cell population. So we will give our object these informations. And yeah, this information is usually from, like, if you're doing experiment, you will know which sample comes from which group. And here we will assign them. But the first thing we will do is, you see the sample names are very long. They have a GSM number at first, and then on the line, and then it's the real sample name, like this one. And what we're gonna do now is to cut off the first part of the sample names to make it more clear here. So sample names. We have we'll create a variable called sample names, and we assign using a function called substring. Is to have an extract the substring of something, and what we're gonna extract is from column names X, and what we're gonna extract is from the number 12 character and to the end, which is the number of characters in column names X. Let's run it. Okay, let's see what's sample names. So currently sample names, it looks shorter, right? It's easier to recognize comparing to these long things. Okay, and then we will assign them to our object by using code names x equals to. Other than using this arrow, you can also use equal. They are the same, but usually I prefer to use the arrow, but you can also use the equal. Sample names. Okay. And also then we will assign the group factors. We'll assign the group information of which sample comes from which cell population. Group as factor. Because it's better to have it as factor. And the orders looks like LP. The first one is from LP. The second one is ML. The third one is basal. And then LP. So, and LP is right. And we'll assign them to X by using X dollar samples dollar group equal to group. So this way we assign the group. Oops, sorry, which one? I'm missing a basal sample. Let me check again. Is it correct? Yeah, it's correct. And then we assign the sample value to X samples. And also, as I talked about, our samples are sequenced in 3 different sequencing lanes. Then we will input the lane information, lane factor. Just to repeat. There are 3 lanes, lane 004, 006, and lane 008. And the number of samples from each lane is 342. And as what we did before, we will assign it to X samples by creating a new column in X sample called lane. Okay. And now you can see what does X samples look like. So now for the group, we are not only having all of the ones. Actually, we're having. Now we can know like the first samples coming from LP and the second samples coming from ML. And also we know which lane are the sample sequence. So our sample information are prepared. Okay, and other than sample information, we also need to organize gene information, which is called gene annotation. Because now the genes are we are only knowing the interest ID, and from the ID we don't know which are the exact genes. Because usually we are calling the genes by their symbols. Like there's a famous gene which is related to breast cancer called BRCA1 and BRCA2, BRCA1 and BRCA2, right? And usually we are recognizing the genes by their symbols and not the interest ID or something. So we will try to create a link between the interest ID and also the symbols. And also, if we want to know some other information about the gene, like the full name of the gene, because you know the symbols are usually some short format of the gene name. And if we want know which chromosome does it come from, we can all find it out in gene annotation. And the way we are finding annotation today is using a package called musculus. That's the mouse. So firstly, we will extract gene ID from X by using raw names X, and then we'll create a data frame called genes by using select. This to select information from the musculus database. And gene ID. Just calling telling the function to use gene ID to find our information and columns. And then key type. Type equals to interest ID, so that the function will know we are using interest ID to find out the information. Okay, and let's run this. Yeah, you can see there's one information. Say select returns one to many mapping between keys and columns. That's very normal because sometimes one interest ID can be related to different genes, or to sometimes like if a gene is appearing in different places on the chromosome, like it's having a lot of copies, then it can be returned many mapping because there are a lot of copies, but they're sharing one interest ID. So it's not a one-to-one mapping, and that's not good for our downstream analysis. First of all, let's have a look at genes. Now the data frame has the interest ID, which is coming from our data, and also the symbols related to each ID, and also the chromosome that this gene is on. And it's normal that for some of the genes we don't know which chromosome it's on. Okay, go back to code. And then we will need to delete the duplicates in the dataset in the genes. And what we do now is to only remain the first one that appears in the genes, and delete the others by genes. Okay, the function called duplicate is to find out the duplicates. Interest ID. Okay. So this way we're deleting the duplicate genes information. Let's have a look. Okay, and then we assign genes to X by X genes. And we'll look at X now. So you can see in our object X, there are three different data frames. The first one is called samples, which I just introduced, and the third one is count, which is there's no difference between when I just read it in the data and the third one is called genes. It looks just the same as the genes data frame. It's the gene information. Okay, and now we have both the gene information and the sample information. the information ready, so we can continue going on to our data preprocessing. And the first thing we'll do is to transform from the raw scale. Well, because in RNA-seq, if we want to measure the expression value of the gene, we can look into the counts, but the counts are correlated with the sequencing depth. Like if if all of the genes in sample 1 and sample 2 are all sequenced in the same at the same level, however, the sample 2 are sequenced more depth, so like the total number of counts in the sample 2 is a lot higher than sample 1, then what's in the result, the gene level count in sample 2 is also higher than sample 1 because the total amount of count is more. So this is not fair if we want to compare between different samples. So we'll need the best way to treat them is that we will transform them in some way and we'll do some normalization. So then first thing we'll do is to calculate. We can translate them into a form called CPM. CPM means counts per million, which means for one gene, how many counts is from one gene? If there are totally 1 million reads from this sample. So we are calculating CPM here. CPMX. And we can also calculate in the log transformative way. Use the function called CPM. Log equals true. Sorry, it's equal. So that's the log transformative way. It's easy to calculate. And we can have a look at the distribution of log CPM values. So here you can see, although our samples may be sequenced in different depths, but the CPM are in the similar distribution. And after calculating that, we can fairly compare which gene is higher expressed and which gene is lower expressed. And we can have a look at how many samples are actually not expressed in the data set. We can look by table some exact count. So we can see how many genes actually not expressed in the data set. Yeah, so we have about 27,000 genes, and among all of the genes, more than 5,000 genes are totally not expressed, which means there's no count from this gene in all of these 9 samples. And if we retain these genes in our dataset, it's not very meaningful because in the downstream comparison, it's totally meaningless. That's comparing a lot of zeros, right? And also for some other genes, although they are expressed in a few samples, but their expression levels are too low. So it's not possible for us to compare between them. and also because the variation will be very high, it's not easy to detect the differential expression between them. So, and also when we are remaining them in our data set, it's not good because it will cause the there may be multiple testing problem in the data set. So we will need what we are gonna do is to remove the genes that are lowly expressed. Remove lowly expressed genes. So there's a function in edgeR called filter by expression. We can use it to automatically detect the lowly expressed genes. Like we create expressions some by filter by expr and using x, and we will need to assign group information. And what does this function does is to calculate, like in our sample, in our data set, which each group has 3 samples. So the smallest group is having 3 samples. So then the minimum group size is 3, right? So we want to find genes which are having less than 10 counts from all of these samples from genes which are having at least 10 counts in any 3 samples will be remained here.
Wei, we have a question. So Monet is asking, what does the numbers obtained from summary ACPM represent?
Okay, so I will summaries calculate some summary information, like mean is minimum, and that's the 1/4, which means in all of the data set, in the data, if we order them in a way from the smallest to the largest value, and the value is at its 25, the mean is the smallest, and this one is at the 25% position, and the median is the one in the middle, and mean is the average, and this one is at the 75, like yes, 75% position, and max is the maximum value, and and here is, so it's showing some basic statistics about the how big and or how small is the value. So just briefly have a look around this numbers. We can see all of the data's different samples are having a similar distribution of the values. LCPM is the log transformated CPM value. Okay, and I just said what is does filter by expression does, and now we were using the results by only retaining these genes that filter past the filter, and have something called keep flip dot size equals to false, which means we will calculate the library size again after that. Okay, so let's see how many genes are there remaining in X. So more than 10,000 of genes are already filtered out in the dataset. So yep, so because some more than 10,000 genes are lowly expressed, so we're filtering them out. And if you are interested, I can plot how does it look like before and after the filtering, because the code is quite long, so I will not type them. I just want to show the result. Let me make the change the shared screen. This one. So that's the A panel is the distribution before our filtering. That's the distribution of density of the log CPM of different genes. So before filtering, there is a peak at the very lowly expressed end, right? But after filtering, the peak is not there. So we get rid of the peak, which are maybe not useful. Okay. And what we're gonna do then is to normalize our data. You may be interested why we have already calculated the CPM, but why still do we need to calculate the normalization factor? You may imagine that one case, if we had two samples, sample 1 and sample 2, and in sample 1, all of the gene counts are 1, while in sample 2, all of the gene counts are 2. And so all of the genes, we can say they are expressed at the same level, but the sample 2 is expressed and is sequenced twice the number of total counts from the sample 1, right? And after calculating the CPM value, the value of all genes in sample 1 and sample 2 will all be the same. And that's the good thing. But usually we know the genes are changing their expression in different samples. So for example, all of the gene counts in sample 1 are still all 1, but in sample 2, most of genes are having a count of 2, but for some genes, they are having a count of 10. So like some genes in sample 2 are up-regulated. So then if we calculate the CPM value, then for sample 2, because the library size is bigger, so the CPM value for these genes which have 2 counts will be less. But we will assume that most of genes are not changing their expression between different conditions. So, but if we just calculate CPM, then their value will be different. So that's not good. So we wanna make sure that even if a small amount of genes are changing their expression, but for all of the other genes which are not changed between different conditions, the CPM value will be the same. So we'll need to calculate the normalization factor using the function called calc_norm factors and method. The algorithm we choose is called a TMM. It's some methods in edgeR. And now we can have a look at examples, norm factors. Yeah, so you can see the normalization factors are calculated. They're not all 1. And you can see they are all close to 1, because in this data set, our data are very you may see the distribution of the counts are very similar between different samples. But in some cases, like some data are very extreme, what will happen? So like if we have another, so I will not type the code, but I will show you that if we copy x again, and we assign the normalization factor all to 1, and we make the count of the first the counts from the first sample to its 0.05 times, while for all of the counts from sample 2 to be 5 times bigger. How does it look like? Okay, so here you can see that if we just calculate the log CPM but not using normalization, then the expression the CPM distribution of sample 1 and 2 will be very different than usual. And we know that's not correct because we just manually did something to the data, and it's not very good. Well, if we normalize it, we can only take them to make them together. So you can see after normalization, sorry, because I've assigned the value log CPM, so it's changed now. Actually, after normalization, all of the values are getting very flat, which means the expression which means the CPM is getting to what it should be. So normalization is very useful if our data, if our samples are very different. And after that, we will have make some other plots. Just clear here. We'll do dimensionality reduction. We'll make some plots to show our data. We'll calculate CPM again. CPM is true. And then we will have two plots together. Because the code is long, I will also just copy and paste them. And so what we're doing in the code is here in the we will assign some color scheme by using some kind of package, and we assign the values that we have two different sets of the color scheme for either groups or lengths. And for the first plot, we will plot for using the groups as the label, and also color them using the color scheme for group. And for the second plot, we will use the color scheme for length. Why does it look like this? It's not. Maybe something's wrong with my laptop. Oh, I will make the plot one by one. So the first one for object. I missed something. So here so here are two plots. We call them MDS plots, and you may find the dots position is different because in the first plot we are using dimension 1 and 2, while in the second plot we are using dimension 3 and 4. And let me explain what does this plot does. Let's consider a situation that we have 3 different samples, and in each sample we only have 4 genes. We only have 3 genes, and if we have a 3-dimensional, we can put them in a space that the x, y, and z axis, and the value on x, y, and z are the expression value of the gene 1, gene 2, and gene 3. And so we can treat the expression and the profile of each sample as a vector. Can you imagine? So it's a space, 3-dimensional space with some vectors. And if we wanna have found a plane which are 2-dimensional plane, and we can tell the difference between different samples. If we have only 3 samples, it's easy to find the plane because the 3 dots can form a plane, right? And we can find an angle to have the x and y axis, so that the dots on the plane, the position of the dots on the plane, it can in the x-axis, it can be separated the most, which means the most of the variance can be explained on this x-axis, while in y-axis it's a little bit less different, but it's still we can have a lot of difference between the samples. But if now we have 4 samples, and if they cannot be put on one plane, we will need to find out a plane that can express most of the changes, the differences between each vectors. So that's what we do for dimensionality reduction, that we can treat our gene expression data as a high dimensional data, that each gene is a dimension, and each the expression value of the expression profile of each sample is a high dimensional vector, and we will project the high dimension data into some lower dimension data and form like what we do to find a plane in a 3D space. So that in the low dimension, in the lower dimensions, the difference of the data can still be shown as much as possible. So in this, we use some algorithm called MDS. So in this plots called MDS plots, if the different samples are closer to each other, then they are more similar. If they are far away apart from each other, then they are quite different. So here, like in the dimension 1 and 2, we can see sample basal, LP, and ML are all separated, while the difference between LP versus ML is smaller than the difference between basal and the other 2 groups. Yeah, right. And also the difference inside LP or ML is bigger than the difference inside basal. Well, sometimes other than just looking into dimension 1 and 2, we may also check the other dimension to see if there are some effects, like, as I told before, the batch effect. Like here we plot the sequencing lengths in dimension 3 and 4, and we find that actually all of the data are separating by length in dimension 3. So which means the difference of the sequencing lengths are actually having some effect to the gene expression profile we sequenced. So if this is the case, we need to take the sequencing lengths into account in our downstream analysis, because it's actually causing difference of of the expression value. So we will need to remove it, then we can compare the expression between different groups. And so these plots are used for us to check whether there is such a thing, such a batch effect. And also sometimes, like if we are confounded the sample label, like if one LP, like if a sample is actually LP, but we label it as basal, then we can see it on the plot. And if we see this very abnormal thing, like a dot is not in where it are not together, and we may check our experiment records whether it's the right tag or whether we're confounding something. So it's very important to make such plots when we're doing RNA-seq analysis. And also we can guess how many what's the extent we can find the differential expression genes. Like if the data the samples cluster very well into groups and they're separating a lot, then we might find quite a lot of deviations between them. But if the samples are everywhere in the plot and they are not separating very clearly, then we may think that the variance inside of the group is very large and we cannot tell it from the biological difference between the groups. So it's not, and so we may not find very many D genes. And other than plotted in this way, we can use some other function called GLMDS plot. It's actually from a package called Glimma. CPM labels this group name and separation using underline and groups examples. Let's assign the chemistries of different how do we color the cells and groups. And let's run this. It's running a package called Glimma. And well, and you can see how can I share the, and there will be, you can see a web page is opened, right? And the plot is the MDX plot that we're just talking about, and it looks very similar to the one in R. However, the difference is there is a bar plot showing different dimension and the proportion variance is placed, and there are also label group and lane. And there's a table at the sorry at the bottom having the label group and lane information, and there's a search bar. So what's special for this plot is that you can see when we move our mouse to all of the plots, we can actually fit them, and the labels and its value will be shown here. Like also the table, we can click each one, and the dot will be highlighted. And we can also use this to order the table, and we can also use the search bar below, such as if we search basal, and here all the basal things will be shown. And was for this bar plot, actually we can click like this way or this way. So in this way, it's plotting using dimension 3 and 4 in the MDS plot. And if we click this lane, now the dots are colored by lanes but not group. And also we can switch back. So it's a fun thing that you can play with the plot if we use a Glimma plot. And actually then it's still the old version of Glimma. For the new version of Glimma, there are some other kind of plots that are also interactive that you can play with. and you can also insert the new kind of Glimma plot into other documents. So it's more useful. If you're interested, you can have a look at the at the user guide of Glimma. And let's go back to our RStudio. And so we just made an alias plot, and after that we will go to the real differential expression part. So before the part, I wish to have a short break, like another 5 minutes. And if you have any question, you can ask me. And you can type it in the chat and I can answer you. Yeah, 5 minutes
Xueyi,
break.
we have a question.
Yeah, sure.
So Monet is asking, can we use principal component analysis for dimension reduction instead of MGS?
Yeah. Actually, they're doing very similar things. That they're all dimensionality reduction, but they're just different algorithms to calculate the difference, and there are also some other algorithm like TSME is usually used in single-cell RNA-seq. Maybe you have heard of, and UMAP is also widely used in single-cell RNA-seq, and they are all different algorithms. And for MDS, my understanding is that although MDS and PCA are all a linear reduction of dimensionality, while the performance of MDS is usually a little bit stronger, like it can separate the different samples a little bit more than PCA. But if you want to use PCA, it's also fine. And the calculation is, it's very complicated. So if you are interested, you can have a look on your own. Let me drink something. Do you have any other questions? Maybe I can wait for another 1 minute. If not, I will continue to some introduction to linear model and the analysis.
So we have 15 minutes remaining.
Oh. Okay, so now after some data preprocessing, we can go to differential expression analysis for our RNA-seq data. First of all, I would like to introduce our tool Limma. The name of Limma stands for Linear Modeling for Microarrays. But actually, currently, we can also apply our linear model to RNA-seq data. The effects are estimated for parameters specified by design matrix. Like here, we have some data from condition 1 and condition 2, and we can estimate the main expression for samples from condition 1 and condition 2. And the expression of each gene is different, but the same model is fitted to tens of thousands of genes simultaneously. Although in RNA-seq usually we didn't have much replicates for each group, but we can borrow information between different genes. So that's why we should use linear model for the differential expression analysis. Otherwise, if we just do some basic testing just based on the data, but not using the linear model to borrow information between different genes, then because we don't have many replicates, then the variance estimation will not be that good. And okay, then I would like to introduce some basic linear models for you. Considering that we have a data set where we have samples from different ages, and we believe that the expression of genes are having a linear relationship between the expression and the age of the samples, then we can have a model like this. And the beta 0 and beta 1 are the parameters that we wanna estimate. And the covariates here are now quantitative because ages can be quantitative. And here the beta 0 represents the y-intercept. And the beta 1 means the slope. So this kind of model is called a regression model, that we are doing a regression between the age and the expression. Well, sometimes the covariates are some factors, which means they are category, like genotype. It's either wild type or mutant, but they cannot be written in some form of numeric things. And if you are using R, you may be familiar with some data type called factor. And yeah, it's the same thing. And here the model can be written in this way. Expression equals to beta 1 wild type plus beta 2 mutant. Here the wild type and mutant can be either 0 or 1. And beta 1 here represents the mean expression for wild type, and beta 2 represents the mean expression of mutant. So for example, if a sample is from wild type group, then the expression of gene is beta 1, which is the mean expression of a wild type for one time, while the mutant here is 0, so the beta 2 is not added. While for samples from mutant, the expression is beta 2, while the wild type is 0, so it's not added. And this type of model should be called as means model. And also there's another way to represent such cases, like this one. The expression is beta 1 plus beta 2 mutant. And here the beta 1 represents the mean expression for wild type, which is the same as the means model, but here the beta 2 means the mean expression and difference between mutant and wild type. So for samples from the mutant group, the expression is beta 1 plus beta 2. So the value mutant for the samples from the mutant group is 1, while for the samples from the wild type is 0. And this type of model is called mean reference model. And sometimes in our dataset we have some factors that are not of interest. For example, sometimes our data are sequenced in different batches, and there may be some batch effect which can affect the expression of genes, but we don't want to account for them because they're not of biological interest. So we want to remove the effect in our analysis. And for example, if we have samples from two groups, wild type and mutant, and they're sequenced in two batches, then we can write our model like this way. And here the expression is beta 1 wild type at beta 2 mutant, which is the same as our means model. And we will also plus beta 3 batch 2. So here beta 1 and beta 2 are estimated for all of the samples from batch 1, while beta 3 here means the difference between beta 2 and beta 1. And we can also consider that here this part is similar to this part in the mean reference model, and then we add the batches. Or we can also use another way, that expression is beta 1 plus beta 2 mutant at plus beta 3 batch 2, and here the beta 1 means the mean expression of wild type in batch 1, and beta 2 means the difference between mutant and wild type, and the beta 3 is the difference between batch 2 and batch 1. So here's a quiz. If you understand what I talked about, please write out an appropriate model for our dataset. And in our dataset, we have 3 cell populations, basal, LP, and ML, which we can consider there are 3 different groups. And these samples are sequenced in 3 different lanes: lane 004, lane 006, and lane 007. Lane 008. I will give you some time to consider. Now, if you have written out your model, and let's see how the answer is. There are also 2 different ways to present our data. Did you get the right answer? If yes, then let's continue. And I will gonna talk about design matrix. It is something that we use to specify independent variables that have an influence on gene expression, for example, experimental group or batch effects. And design matrix can be specified with or without intercept. And if we have intercept, this is the second way of model that we just explained, while without intercept is the first one we explained before. And here we will focus on intercept-free design matrix in our dataset, while you can also use the design matrix with intercept if you like. And if you want to learn more examples of different design matrices, or if your data are very complex, such as you're having a time course data set, or you want to fit some covariates, then you can have a look at either the Limma user guide or the design matrix and contrast guide paper. And the paper is here. You can access via this link. And in this paper, you can also have a read of some basic explanation of the linear model. Estimate the variability. And if we wanna estimate this, we would need to know the distribution of the data, and then we can calculate the p-value, which are of our interest. Okay, and when we write out the design and contrast matrices, it looks like this. For design matrix, you may find there are quite a lot of ones and zeros here, and each column are associated with model parameters. Like B1 is associated the parameter beta 1, and B2 is associated with the parameter beta 2, and the rows are associated with samples. For example, for sample 1, the expression of genes in sample 1 is 1 times beta 1 plus 0 times beta 2, so it's just beta 1. While for sample 5, the expression is beta 2. And although in our situation, all of these the numbers in design matrix are either 0 or 1, but in some cases it can also be some other numbers like 0.5 or some other things. And if you're interested, you can also read the design matrix and the contrast guide paper. And in contrast matrix, the columns represent the contrast of interest, and the rows are associated with model parameter. So for example, for the contrast C1, it is B1 is the difference between B1 and B2. So can you understand this? For you to understand it better, I can write out the model for here. So let me show you the design matrix for our data. This is the code to generate design matrix. We use the function called model.matrix, and this 0 plus means we are removing the intercept in the model. So the design matrix looks like this. So for example, in sample 1, it's coming from LP and it's from lane L004. So in its design matrix, its expression is coming from the main expression from LP from lane 04. While for example, for sample 7, the main expression of genes in sample 7 is the main expression from sample from basal samples in lane L004 plus the difference between L006 and L004. Since this sample is coming from basal and it's sequenced in lane L006. And then we will also set up the contrasts. Here we want to make a pairwise comparison for all of so our contrast matrix looks like this. We compare all pairs of the different sample groups, and the contrast looks like this way. You can see all of the lane L006 and lane L008 is 0, which means in the contrast we are not taking account of the lane information, so the batch effect is removed. And let's go to some information about the linear model, because in RNA-seq data, our data is discrete, which means that our data is coming in the form of counts. It's either 0, 1, 2, 3, 4 or something, and it cannot be like 0.1 or 10.5 or something else. And also it has a non-constant mean variance trend. Well, as I've told, our model Limma was originally designed for microarray analysis, and the microarray data follows normal distribution. However, our RNA-seq data is not following normal distribution. So here we can use a function called voom to firstly transform our counts to log counts per million, log CPM. So our data can be continuous. And also by using voom, the mean variance trend can be removed by using precision weights. So after running voom, we can assume our data follows normal distribution. And then we can apply the limma model to our RNA-seq data. So here, like this way, before using voom, you can see there's a trend between the mean expression of genes and the variance of the gene. And in this plot, each dot means a gene. And you can see there's a trend. And by using voom, we can calculate a gene-specific weight by fitting the trend. And you can see after applying the weights calculated by voom, the mean expression and the variance trend is removed. So our data is following normal distribution. And that's how we use voom. Just use the function voom, and we assign the design, and we set plot equals true to see our plot. And let's see what's inside of V. In our voom results, the gene information is contained, and the targets means the sample information, and E here means the log CPM value calculated, and here you see the weights is the voom precision weight. And after using voom, we can fit the linear model. We use the function lmfit. That's fitting a linear model for each gene, and we fit our contrast. That's to compute statistics and fold changes for our comparisons. And then we use a function called eBase to compute more precise estimates by sharing gene information using empirical Bayes moderation. And then we plot the residual standard deviation versus the average log expression for the fitting model. And that the plot to showing you that the mean variance trend is removed in the final model. And why do we need to share gene information in our RNA-seq analysis? That's because our experiments have a very small sample size. Because usually as the sequencing cost is very high, and also our amount of the RNA sample, the material is limited. So usually we don't have like 10 or 20 samples in each group. So if we have a small sample size, then the estimation of variability within an individual gene is very unreliable. So what we do is to pull in information from all genes, which can increase the statistical power. And what does the empirical based method do is to borrow information from all data to form a prior information that is used to adjust the observed value to values that are more precise. And this is the way we do in Limma to calculate a posterior variance. And the moderated t-statistic is also calculated using some posterior variance. And this may not be easy to understand, but I will show you some plot. That's the variability measurement before and after applying E-Bayes method, and each dot here in the plot means a gene. And you can see after applying the method, the variability is more closer to the line, which means the prior information. And after that, we can test for differential expression. We use the function decidTest to test DE, and that's the result. We can see the number of D genes between basal and LP or basal and ML is much more than it between LP and ML. And you may remember that previously in the dimensionality reduction part in the MDS plot, the basal group is much far away from LP or ML. And yeah, the result is similar to what we considered before, right? So the decidTest function is to identify genes that are differentially expressed and also adjust the p-value. Here we shouldn't use the raw p-value because there is like for each gene we are doing a test, and if we're doing quite a lot of tests at the same time, then the possibility of having more than one false positive is getting higher. So to control the false positive rate, we have to adjust the p-value for the multiple testing. So when we wanna see the differential expression result, we have to see the adjusted p-value but not the raw p-value. And the summary function is from basal, and we use it to summarize the number of differentially expressed genes for each comparison. And the first row, the first column means the comparison of expression level between basal and LP, and the second one is between basal and ML, and the third one is between LP and ML, which is the contrast we said before. And the first row means the number of downregulated genes in basal in the first group like basal when we compare to LP or the other groups. And the number means the gene that have significantly lower expression. And the significance is identified by adjusted p-value less than 0.05. And also similar to this, the third row means the genes that are having significantly higher expression. And if we want to have a look at the top, what exact genes are differentially expressed, we can use a function called top table. That is to list a table for any of the comparison. Like here, if we want to see the top 10 most significant differentially expressed genes for the basal versus LP, we can use top table, E-fit, coefficient is basal versus LP, and that's the result. So here the log FC means how big the relevance difference is comparing basal to LP, which means the main expression of basal, how many times the difference between the main expression in basal comparing to the main expression to LP, and then we log transform the fold change. And this column means the average expression across all samples of this gene. And here the t, p-value, and just p-value are our results from the linear model, where t is the t-statistic from our t-test, and adjusted p-value is the most interesting value which we want to look at. And sometimes, like in our dataset, we find that the number of differentially expressed gene is quite large, and we want to do something which can reduce the number of D gene, and also we want to test the D relatives to a threshold, which means that if we want to find out D gene which are only having like the difference is more than 2 times between the comparison, then we can use the method treat. It's to test for D for a minimum fold change, where the difference that are large enough can maybe more biologically meaningful. And that's the way we use it. We use the function treat, and we set the log fold change of our interest to be 1, which means our minimum log fold change to identify D genes is 1. And then we also run the design tests and run summary, and here that's the results. We can see the number of D genes is reduced. And we can have a look at the number of D genes in common across 2 comparisons, because sometimes, like if different comparisons are having some biologically link, then we can use like in this way. So there are 2,700 common D genes between basal versus LP and basal versus ML. And we can have a look at the top 20 common D genes here. And also we can make a Venn diagram in this way. And we can also use the function write_fit to write our model fitting results into a file, so you can see the file like in your interested way, like open it in Excel and explore the results. And as similar to the top table function, if we want to have a look at the top genes from the treat results, we can use a function called top treats. And if we set n, which means the number of genes we wanna pull out from the result, if we set it as infinite, then we can pull out the full results from our DE tests. And sometimes we can write it into a table, which you can also explain and explore it in some other software you like. And let's have a look at the top. Looks like this, and it's similar to the top table. It's also having the gene information and the log fold change and the average expression among all of the samples, and also t-statistic and raw p-value and adjusted p-value. And we can have a mean difference plot using this function to see the results in a whole. So the plot may look like this. Each dot represents a gene, and the red genes are all of the upregulated genes, and the green ones are all of the downregulated genes in our comparison. And also we can have an interactive version of the MD plot using the Glimma package in this way. And if we set launch as true, then we can directly open the results. And the interactive MD plot looks like this. This part is similar to the MD plot we've looked at, and all of the dots can be clicked, and the table, everything can be clicked, and you can search it. And here, this panel shows the expression value of this gene in each individual samples. So if you want to send your results to the person who are doing the biology, then you can make a Glimma plot, and if you send them the link, send them the website, then they can play with the results by themselves, and they can explore each gene they're interested in, which may be very useful if you want to collaborate with someone else. And we can also have a heat map. Here, let's show you how to make a heat map of top 100 genes for basal versus LP comparison. The code, firstly we will extract which are the top 100 genes between the comparison, and we get which row are they in the data structure, and we assign some color, and then we make the heat map. It looks like this. So here each column means a sample, and each row is a gene from the top 100 genes. And if the samples are more similar to each one, they will be closer on the heat map because it has been clustered. And you can see that the whole trend. So you can see the basic pattern of each gene expression, and if the gene expression profile is more similar for any two genes, they will be placed closer on the plot, so which may be of interesting. And that's all we're gonna show. And if you are interested to look into some more details, you may go to these papers here. And thank you for attending. I would like to thank Matt Ritchie and Charity Law, which are my supervisors, and also other members from Ritchie Lab. And I would like to thank Gordon and Connie who have helped with this workshop. And I will also thank Hidia and Muna from Our Ladies Tunis who are organizing this event, and also thank to Our Ladies Melbourne, which is the local organization in Melbourne, and I have attended several events, and it's a great organization.
Thank you very much for attending. We'll have another R for Bioinfo session on the February 17th, and it will be about data integration. So it will be about how to integrate a microarray with ChIP-seq. Thank you everyone for attending. We'll send you all the slides, the code, and the recording. Thank you everyone. Thank you, Xueyi, for being part of this workshop and for accepting our invitation. And please stay tuned about our events. Thank you very much. Thank you. Bye-bye.